Okay, hi everybody. Thanks to come to the next seminar of the IATA Institute. And we are in a very special day because we have someone, very relevant person in the area of synthetic biology today. He is professor in the Bioengineering department of the Imperial College London. He has more than 60 publications in outstanding journals like Nature Biotechnology or Cell. And as a funny story, he participated in the human sequencing project when he was only 16 years old. Maybe he can comment on that later. And the Tom Labs participates in the Synthetic Yeast Genome project, maybe one of the biggest synthetic biology projects in the world. And today he's going to talk some things that they do in their lab related to the synthetic biology in yeast in the production of different raw components, new materials. So thanks, Tom, to come, and you can start.
Great. Thank you very much for the nice intro. Yeah.
Okay.
So you can, let me get rid of that. Yeah, so you can see my slide here, and this is where I am right now at Imperial College London, in central London, just next to Kensington Gardens, where there's some nice royal palaces, but also nice gardens for us to walk around. Although I obviously spend most of my time stuck here in my office. I think it's just somewhere around here, if you can see my cursor. Do you see my cursor moving? The arrow, do you see the, no, yes. Maybe not. Yeah, okay, just want to check you see that. Yeah, so we're over here, yeah. But yeah, when I was a teenager, I lived in Cambridge, and near my house was where they'd started doing the human genome sequencing project. And so...
you just jumped there.
Well, my friend said, oh, I've got a job at this place through that I heard they need people to load DNA sequencing gels at the weekend, and you get paid per gel that you load. It's fun. It's kind of weird, and we can play computer games.
Wow.
on the network system, Doom, the famous Doom. So I used to load a load of gels and then play Doom until they finished running, and then analyze them on Saturdays and Sundays in this big building, old building at the time. There's a new one now. And no one else was around because it was the weekends, and I was 16, and I got three pounds for every gel I loaded.
Okay.
A lot of wells though, 90 wells. They were quite complex to load. Wow. And so it was good pay. What's funny to think is that those were the only gels that got loaded for the Human Genome Project on Saturdays and Sundays, which is 2 out of 7 of the days of the week. So that means for that year, 1995, I think it was, I was responsible for 2 out of 5, so like 20-something% of the UK's contribution to the Human Genome Project.
Wow, the world owe you a lot about that.
Yeah. Now, during the week, they ran many more gels. They just would run a few at the weekend, and they were me. Anyway. Right. So let's get on with this. Synthetic biology to grow materials. That's the topic. We do a lot of different things in my group. All of them at the moment in microbes, although we are moving into human cell stuff as well now with the Sanger Institute, which is where I used to be based doing the Human Genome Project. What we do, let me see if I can get the slides to move, is we basically do what a lot of other people do in the field, which is we try to treat a microbial cell as something that can be reprogrammed to do tasks for us. And we try to think about it a bit as like a machine that can be engineered. So I like to tell my students that I teach that synthetic biology is the idea of engineering cells to become new technologies. So we do this by applying some of the things we see in other areas of engineering, principles like modularity, measurement, tools from computer-aided design, modeling-based predictions, to engineer biology. to get these cells to do things for us, hopefully programming them in the same sort of way that you might program a computer. But ultimately, the way we really do it in our lab, and what I probably tell my students, particularly the undergraduates, when I'm telling them what we do, is we basically reprogram cells by treating DNA as reprogrammable molecular code. It's obviously an analogy, it's not the reality, that DNA is like the software for an operating system, but it's a good analogy because really that, I think, helps you think about what we do in synthetic biology, that within each cell, there's this operating system, the genome, and we can go into that, edit it to get different operations to run at different times. Or we can add in new DNA encoded programs that get the cell to do some task it wouldn't normally do. And that's kind of quite similar to someone who writes an app for something like your smartphone. You know, it's a reasonably small amount of code that can be downloaded onto your phone and give your phone some new ability that was not written into the operating system. So those are the sort of things us and pretty much everyone else in the main synthetic biology field does, is basically taking DNA code and going, okay, how can we reprogram this, rewrite this, move modules around to get the cell to do something different and interesting. Cells, the different strains we work with, are basically our different operating systems. And so these are the favorite operating systems of my group. Saccharomyces cerevisiae yeast. I have a cuddly version here behind me with its little buds, you can see. E. coli bacteria, the most popular thing for molecular biologists to work with everywhere in the world. And we've also dabbled with a few other things, Geobacillus, a thermophile, Bacillus subtilis, a bacteria associated with food production that's very good at protein secretion. And similarly, Pichia pastoris yeast, a yeast associated with animal food production that is very good at protein secretion as well. And one we moved into in the last 5 years or so that I'll talk about quite a bit today, along with stuff on yeast, is this K. raticus bacteria. So I'll come to that in a minute, and that's something about half of my group are focusing on these days. What do we do in synthetic biology? What are we coding in my group? Well, we work in loads of different areas. Fundamentally, the idea that you can reprogram DNA code to get cells to do new things is a technology that can be applied very broadly, right? Whatever cells do, however we interact with cells, and however we want to get living things to do stuff for us, there's an application of synthetic biology in that area: healthcare, materials production, food production, bioremediation. Areas in particular that my group have focused on over the years: optimal use of cell resources to try and be more efficient in the way we engineer cells to act as machines for us; building synthetic genomes. I won't talk about it today, but we're part of this international project to synthesize redesigned versions of all of the yeast chromosomes to make a synthetic yeast genome. And using these sort of tools of synthetic biology, particularly modularity, to optimize and explore pathways, to tune cells to be better at running pathways. And when I say pathways, we've worked on two completely different types of pathways. We've worked on pathways of enzymes that run metabolic reactions to be able to get cells like yeast cells to produce complex molecules like antibiotics. We had a project where Baker's yeast was used to engineer penicillin biosynthesis and secrete the antibiotic and kill bacteria. But we've also tuned signaling pathways, and our publication a couple of years ago in Cell looked at how you could tune the sensing of things through G protein coupled receptor sensors in the outer surface of yeast cells and translate that information to sensing changes and actions inside the cell. But today I'm going to focus on functional materials and our work in that space, which is somewhere where we've had some recent good papers and lots of nice progress. And it's also relevant for you guys because it's all stuff from food. And we're using that now to make materials. So here we are, materials. I'm going to start with a bit of introduction about why I'm so fascinated with materials and synthetic biology, and why I think synthetic biology and materials is a really great fit. This is an electron micrograph of the cellulose produced by bacteria with the cells trapped within it. This material here is cellulose almost totally, along with just some bits of bacteria as well. And cellulose is, and we always have to say this in the field, the most abundant polymer in the world. And here is an example of somewhere where you see a lot of cellulose in a wood, in a forest, right? And what's remarkable about the plant kingdom, particularly if you think about walking through a forest, is you have these huge organisms, which are predominantly made of cellulose, trees, but they have different material fields and different properties in different places in that tree, right? So you'll see the leaves are flexible, you'll see the wood. The wood will have different properties at different parts of the tree. So the wood in the roots is more flexible, the wood in the branches is less flexible, the trunk is much more solid. But then other areas like the leaves, petals, when there's flowers, the seed shells, all of these are very different material properties, okay? And so you can even see a few here, different material properties. You know what it's like to go touch things in a wood, so you can imagine what these things feel like. But each one of these trees with its plethora of different material properties, every has been produced from a single cell with the same genome. And so all throughout the this big tree organism, every cell contains the same genome. But as the cells differentiate, different regulatory programs turn on in the cells that instruct that cell and its neighbors to make a different thing, which has a different material property. And that's really exciting, I think, for synthetic biology, because that shows us from the plant kingdom that there is this ability to encode in cells the information to define the material properties of something that's made. And you could switch on and off genes in different cells in these programs and have cascades of differentiation to make different programs, and then have your one sort of program cell go off and make different materials to design with different material properties. And that's something I think synthetic biology should be ambitious enough to be going for. And it's actually something I think synthetic biology is very smart to go for, because it allows us to use lots of the tools that we are developing in synthetic biology. So how does biology produce different material properties in different parts of something like a tree? Well, generally in the plant kingdom, the different material properties come from two things. So every cell grows and has cellulose around it. That's pretty much universal. But then the material property of that tissue with cellulose changes based on composites, so interweaving different biological polymers around the cell with the cellulose, so things like pectin, glycans, and then of course things like lignin and xylin as well, all sort of adding to the material properties by putting different things amongst the cellulose fibers. And then the defined spatial arrangements. How do the cells link with other cells? How much air is in the cells? So they'll have those big air vacuoles. How much water is there? Those sort of things also really help change the material properties from a dense wood to a very light, flexible wood. And so all of these things are genetically programmed. And all of these things, we hope, are things we can understand how to genetically program. So we can write DNA code that says, I want cellulose plus these composites, and I want in these arrangements, and therefore that will give me this kind of material that I can then use in applications, in healthcare, in building things, in all sorts of different areas, in clothing, for example, as well, right? Clearly, the whole world is full, our world is full of things that we get from the plant kingdom that we use as materials, from the wood desk behind me to the clothes I'm wearing, and many other things as well. So if you want to learn how we can write these DNA programs to grow materials to design, well, we have tools for saying, okay, I want to make this chemical and I want to make this polymer in synthetic biology. And that gives you the composites. You can say, okay, turn on the production of glycans, turn on the production of a polymer like spider silk, for example. Some people have engineered cells to make spider silk. And then that mixed with the cellulose that's also been made will start to give you polymers. And then defined spatial arrangements is something as well that synthetic biology has been going for years. There are many groups who have been, for example, pursuing DNA-encoded programs that can get cells, mammalian cells, yeast cells, and bacterial cells to do things like form Turing patterns, where they will self-assemble to make circles and stripes and things like that. And so that would be a powerful set of tools if you're building materials and you want to do spatial arrangements. So if we want to learn how to do this, there's basically two ways. One is you can take a plant genome and you can find all of these DNA programs and try to reprogram them and strip them down and work out how they work and then rebuild them back up in a way where we have control of. That is a nice idea. It is difficult to do because a plant can't be a plant and survive unless it can do lots of different material properties at different times in its life cycle, right? It needs to start as a seed and grow and make leaves and things. So if you disrupt material property encoding programs, you don't have a viable plant. So if you really want to do this in a sort of more systematic way, you need to start with something much more simplistic, something like what we say in English language, the phrase is a blank piece of paper. And so we have done that, and our blank slate, our blank piece of paper is bacterial cellulose, a much more simplistic material than the cellulose we see in plants. It is grown by certain types of bacteria, which will ferment sugar over just a few days in a stationary growth as long as oxygen is present, and they will produce this biofilm layer that we call a pellicle that will float at the surface at the air-water interface, and it is just predominantly, ultra pure cellulose. None of these other polymers mixed in. Okay, so this is one of these pellicles. You can see it here from above. It's just a big, kind of almost like a hydrogel of ultra pure cellulose, glucose repeated many, many times. And so we think this is, it literally is like paper, right? So it's just cellulose. It's a blank piece of paper we can learn how to modify. So here's some of the biology of these amazing bacteria that produce it. So we use one that we've isolated ourselves from kombucha tea, from a kit for making your own kombucha. We isolated this bacteria. It's called Kombucha raticus. And it's part of the acetic acid family of bacteria. They like to grow on sugary water at a lower pH, and the majority of this family will produce acidic growth media, which we use as vinegar all around the world. And then some of them will also produce this material layer on top, and we call them the Kombucha or the Glucona Acetobacters. And that material is sometimes used in the food industry to make a dessert that's popular in Southeast Asia called nata de coco, a kind of a cellulose-based jelly with sugary coconut water in it. And it's also a byproduct when people are making kombucha tea. They get the material layer on top and they throw it away or save it to then set up the next fermentation, and they only take the liquid from below as their drink. Anyway, these bacteria have, for some reason, evolved to take about 30% of all of the sugar you give them and immediately polymerize it out by this mechanism through the BCS-AB polymerization system and then out through the CD pore and just put out cellulose. They don't have the machinery to then break down that cellulose and eat it again, so they're literally just making a material that they don't ever intend to digest again, with a third of all the sugar that they get, which is kind of bizarre, but somehow it's part of their lifestyle. It is threaded from the individual polymers out through the membranes and starts to self-assemble and thread together into these larger fibers. You can see this beautiful electron micrograph from a group in Japan. I can show some of these fibers coming out as these bands, intriguingly all from one side of the bacteria. And then after a certain point, maybe through motions and things like that, these collapse on themselves and become these thicker fibers, which are kind of similar to the sort of fibers you get in clothing or in paper if you were to look at these things under an electron micrograph. We see this same amazing behavior in our lab. This is a microfluidics time lapse of our bacteria producing cellulose stained in blue. And you can see it all comes out from one side of the bacteria in these bands. You can even see as the cells grow and divide, these ribbons start splitting up. And then if this gets shaken about a bit, they start to, these sort of waves of cellulose become these long, thicker fibers. If you take the material after it's grown, dehydrate it, and look at it under an electron micrograph, it looks like this interconnected mesh with some of the bacteria still trapped within it. And it's interesting to look at this because this is a very, very tight interconnected mesh. But another thing that's worth noticing about this electron micrograph is the fibers are much longer than the cells. They are extruded from the cells get pushed around in the process of these things getting made. And so some of these fibers can be, you know, not just microns in length, but millimeters in length. And potentially, theoretically, nothing's to stop it being the full distance of your tube or your flask that you're growing in. And that actually means it has a very good material properties compared to cellulose from plant tissues, where the cellulose is only ever coating the individual cell. It doesn't disappear off and go over and connect with other cells microns away. It will surround only its own cell and maybe intermingle a bit with the neighboring cell. With this, it goes all over the place. And so it connects into a very tight mesh. And that means it has really impressive material properties, much better material properties for materials nerds than plant cellulose. And so because of that, it's used in many products. It's already used in healthcare and cosmetics. It's used in the acoustics industry for very good acoustics in headphones, for example, expensive ones. And Samsung have been developing it as a separator between different parts of their batteries to try and be able to bring down the weight of the batteries whilst increasing the capacity. This is a slide my collaborator who works in material science presents, showing that you can just get your bacteria, grow the medium, get your culture after only a few days, and you get these fantastic material properties out of it. What drives these really good material properties out of it is just really the uniformity, the sameness of the material. It is just glucose, 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 glucose in huge long polymers with very little branching, no other polymers are present, no contaminants, and very long scales of this being produced and coming together into these fibers. I was trained as a molecular biologist and biochemist. So for me, frankly, cellulose is kind of the most boring molecule there is because it's just glucose forever. But for materials people, that's what makes it super special. So I'll tell you a bit about our work in engineering this bacterial cellulose using synthetic biology. And then I'll talk about bringing that together with yeast, the other thing we engineer, to be able to make materials that can do stuff in cool ways. We got into bacterial cellulose actually through an undergraduate competition called iGEM. We had a summer in 2014 where we had a fantastic set of undergraduate students supervised by my PhD student at the time, Ben Reeve, who spotted that we had two materials experts at Imperial, Alex Bismarck and Kunyang Li, who knew about working with bacterial cellulose, and they'd previously collaborated with a famous biodesign artist, Suzanne Lee, to grow the bacteria actually in her bathtub and use it to make materials as a leather replacement. They spoke to these people early on without me being involved and came up with the idea that, well, maybe we can change people to working with these materials for clothes and for other things if we can add synthetic biology so that we can make the materials do special cool things that normal materials like leather don't do. They had a very successful summer iGEM project, amazing students. They isolated the bacteria from kombucha to work with it. And by combining a synthetic biology standard called SIVA plasmids from Spain, from Victor La Lorenzo's group, with the BioBrick cloning standard used in iGEM, they were able to get to the point where they could start doing synthetic biology in this bacteria that they'd isolated from kombucha. Their project aim was to grow this into material layers that can have functional, like peptide things expressed on the cellulose that allow special filtration of water to get contaminants out of the water. And so they called their project Aquilose, with the idea of doing custom ultrafiltration membranes grown from bacterial cellulose with proteins expressed and put onto the cellulose that can then bind specific contaminants. We were runner-up in the world as the iGEM project that summer, but we went on to really make the most of this project. So the core of the students founded a little startup company called Custom M on this idea, where the cellulose now in pellet form is modified with not proteins now, but with peptides that can bind contaminants. They targeted this contaminant PFAS, which is known as a forever molecule. It's a terrible pollutant present in groundwater, very hard to get rid of. And they've become very successful in this. The company got rebranded as Pure Affinity. They've grown now to, I think, 10 to 20 employees and have a market valuation up to 10 million, which is amazing for a project started by undergraduate students only 5 years ago. One of the students who was less inclined to go into industry, Michael Floria, stayed in my group just for 1 more year. And as he was finishing his undergraduate degree, he did some amazing work putting in these DNA components that people normally use in synthetic biology in E. coli and testing them whether they could work in our now isolated Kombucha reticus cellulose producing strain. And this helped us build up a genetic toolkit of little bits of DNA program we can put into plasmids and put into these bacteria to get the bacteria to respond to things and do things. So he developed promoter systems, managed to get GFP and RFP fluorescent proteins working in the cells, and developed inducible promoter systems so that the cells, when growing the material, would respond to chemicals that we give and then start to express genes. And so you can see here we are growing the material, and halfway through the 3 days of growing the material, we start to add a chemical known as AHL, a signaling molecule. And then that triggers that the cells, as they continue to produce the material, they now also produce red protein, red fluorescent protein, and we now get this pink layer being produced in the latter stages. And if you zoom in on a micrograph, you can see the red fluorescence all dotted inside the cells of the bacteria within the material. Michael even went as far as isolating the genome and sequencing the genome of this organism, all while still an undergraduate project student. And he was able to get a publication in PNAS describing all of this with the rest of the team, and another one in another journal as well. And this was really an amazing, I mean, he's an amazing student. He's off at Harvard doing his PhD now. It brought in, obviously, a whole lot of extra funding and enabled us to beef up our team. So in the last 5 years since then, we've been working on this strain and helping expand the synthetic biology toolkit to get this kind of kombucha bacteria to make different interesting materials for us. The things we want to achieve is this DNA encoded programming to get new material properties. So we want a toolkit that allows that, and we want that toolkit to be able to do self-organization of structure, be able to do composites with biopolymers, and to be able to produce and hopefully also secrete enzymes that then can modify the polymers to give different material properties. This work has been predominantly led by Viv Hussens in my group, a postdoc who's just recently left us for a company, and Marcus Walker, who's done this work over his PhD. So the first thing Marcus did in his PhD was to follow up from Michael's work and give us an even better version of the genome. So we used nanopore sequencing to make our final version of the genome with its multiple plasmids. It's a little bit of a complex bacteria compared to E. coli. And that also enabled us to better understand where it lies among the Acetobacter bacteria in its phylogeny. We found actually that it's almost identical to this K. xylanus strain used in China, where also some interesting synthetic biology and engineering applications are being used. We would suggest that they change their name, considering their strain fits so perfectly in with the reticus group like ours does as well. In fact, we think it's almost identical strain. We've also done RNA sequencing of our bacteria, both in growing, shaking in liquid, and then also when it's within the material growing. And this is interesting for us because it enables us to identify genes which are differentially expressed in the different conditions of whether you're shaking and not producing a material layer, or whether you're leaving things to sit still and the material layer is happening. And so these allow us to then identify genes and therefore identify promoters that are specific for turning on and off in those different conditions. So they're very useful tools for us. We have spent a lot of time and effort, and we are writing the paper now to describe this to people, to build up a toolkit that fits in with the Golden Gate standard of synthetic biology molecular assembly of DNA parts, because that is really the one that works the best and fastest for people. And so we've built this KTK Golden Gate toolkit, which we are hoping to publish and share with people by the end of this year, which has lots of different DNA parts, which can be very quickly assembled together to make programs that you can put into these bacteria and get them to do new tasks. One of the first things we've done with that, and we have already published on this a couple of years ago, is get the cells to do the beginnings of being able to form patterns. And so this requires, if you want cells to be able to work out what other cells are around one another and then change their behavior to be able to, turn on and off material production in patterns, you need cells to be able to communicate from one another. And this is well known for people who are trying to do things like Turing patterns in E. coli. You need cell-to-cell signaling, and that is normally done through something that is called in the bacterial world quorum sensing molecules. And the key quorum sensing molecule people like to use is AHL, acyl homoserine lactone, and that's the one we've used here as well. So we engineer one bit of DNA program, we write it and assemble it with Golden Gate assembly. That puts inside one of the bacteria this plasmid that leads to continual expression of an enzyme that produces this homoserine lactone signaling molecule, and it diffuses out of those cells. And so the cell that gets this plasmid we call the sender cell, because it's sending out this communication signal. We then produce with our toolkit a second plasmid which contains a couple of different genes. One of those expresses a transcription factor which, when it binds to this chemical, will then go and turn on expression of another promoter and then can switch on a gene in response. The cells that get this plasmid are the receiver cells because they receive a signal and then act upon it. Okay, and in the case we published here, we have them simply producing red fluorescent protein in response. And so you now have two bacteria that they both grow and they both produce material, but they also signal to one another and turn on and off red fluorescent protein production depending on the signal and the density and how close they are from a receiver cell and a sender cell. And so we can show that you get material produced with red fluorescence only if you get both of these strains together. The receiver can't produce the red fluorescence unless the sender is sending the signal, so you need the two mixed together. And so this is great. This allows you now have ways to be able to engineer boundaries within a growing material where cells produce something else other than just the cellulose. And in the system we have here, we grow sender cells and receiver cells separately for a couple of days, cut fragments of the material that have been produced, now put them close to each other and allow the growth to continue. And that will lead to these bits of material growing further, now fused together. And the gene expression, in this case with red fluorescence, only occurs at that boundary. And so that is the beginnings of being able to allow the kind of things we want to achieve with pattern formation during this material production. So for example, you could say, let's in the future swap out this red fluorescent protein production for something like production of a protein that is like a molecular superglue, which helps to link these different pieces of cellulose together with stronger capacity. Other tools we've developed for our toolkit, CRISPR-based silencing. So we use the deactivated version of the CRISPR enzyme Cas9. And if you provide specific guide RNAs, you can now turn down genes, silence genes within our cells. And others in the field have started to join us with working in synthetic biology in these bacteria. A team from Singapore expanded our toolkit, showed our tools also worked in other Acetobacter that are used in cellulose biosynthesis productions, and also showed CRISPR working. And a very famous metabolic engineering group in Korea, led by Sang-Yup Lee, who's a major figure in the World Economic Forum. They showed as well that you can do significant metabolic engineering of these bacteria to be able to get more cellulose produced via tweaking enzyme pathways. And they did that work with Samsung for their battery separator project. One thing we've done, which is a bit more fun recently, but also quite relevant for things like future materials in clothing and fashion industry, is get ourselves to express enzymes which change the color and to an extent as well the texture of the cellulose that gets made. And so Marcus has enabled, through expression of a tyrosinase enzyme, a cascade will occur in the growth of the bacterial cellulose that also leads to the pigment polymer molecule melanin being produced. And melanin is the molecule that gives us UV protection in our skin and is involved in tanning. And it's also present in lots of different organisms around nature. So, you know, it's a key molecule used by biology for giving a black color, but also giving certain material properties and also UV protection. And so it's fantastic when we first tried to express this in our bacteria, because it was one of these kind of experiments where the result was very obvious very quickly. Here it is. You got black cellulose. And if you like black as a color, you're one of these people who maybe like black clothes and black music and things like that. Well, this is really, really black cellulose when we make it. It's very stunning. And so we're looking to commercialize this as well. There's lots of people interested in this in the fashion industry. We have tried composites as well. This is a little bit harder because our bacteria are very proficient at secreting a carbohydrate, cellulose. They spend their life just churning out huge amounts of cellulose. The composites we're interested in are composites of the cellulose plus some other biopolymer. And in particular, we'd like it to be a protein-based biopolymer, because with proteins, you can start fusing other bits of proteins onto them and get really cool things. So in synthetic biology, the best, sort of most advanced living materials people have done with protein polymers have been ones in E. coli with a type of amyloid protein called the curly fiber. And so the curly fiber system is expressed from the CSG operon in E. coli. And this expresses a whole complex that sits in the outer membrane that exports out a protein called CSGA, which self-polymerizes to produce these very tough, very adhesive threads of protein, these amyloid curly fibers, which allow E. coli to stick in the gut in pathogenic E. coli. And so people like Neil Joshi and Tim Liu with groups at Harvard University have been engineering E. coli to express curly fibers fused to different things, and they've shown some really cool materials they can do with that, albeit at very small scale. And so we thought we would like to be able to try and import this system into our bacteria so we can get our bacteria to churn out cellulose and at the same time produce some of these exotic adhesive proteins as well to make composites. This has been very challenging. We have managed to achieve it. Curly is secreted. We can detect gold particles binding to curly histag, but the curly alone is not really made in anywhere near the yields that are required to change the material properties. And part of that is because our cells are very poor at protein secretion and incredibly good at carbohydrate secretion. So the scales of how much protein versus cellulose being produced is just ridiculous. We're having hundreds of thousands times the scale of cellulose compared to protein. And so it really doesn't change the material properties enough once we start making these proteins. So just to summarize here with K. raticus, we've sequenced the genome, sequenced the RNA, we've got a Golden Gate toolkit working, we have inducible promoters, CRISPR, cell-to-cell signaling. This is allowing us to do things like self-patterning within the materials, expressing enzymes that run reactions that can change the material properties and the look and the feel. And we've shown that we can do some protein secretion, but we really need to improve on this. And we have lots of people in the group right now doing protein secretion as their little project to see if we can get that solved through other ways. And of course, we have other people around the world working on these problems as well. But I mentioned earlier, we also work with yeast, and yeast has given us a way to cheat this protein problem. Okay, and the inspiration for this comes from food and drink industry. It comes from kombucha, which is where we originally isolated our bacteria from. My PhD student from a few years ago, Charlie Gilbert, along with a PhD student in Tim Liu's lab at MIT in Boston, USA, thought about this problem and realized that kombucha, which is incredibly popular in recent years, particularly in the US, in places like Berkeley here. This is a photo I took when I was in California of an entire row of a supermarket dedicated only for kombucha. Kombucha is this, it's not just our bacteria, this acetobacteria. There are other things in a kombucha fermentation, and some of those are yeast. And that's reasonably well known. When people are making kombucha at home and growing it, they talk a lot about a SCOBY. They sometimes call it a mother. And this is what they think is, they call the SCOBY, or the mother, this jelly thing that we have been calling a pellicle and saying this is a cellulose material. They just see it as this bit of jelly that they use, and it sets off the fermentation of the kombucha underneath in the jam jar or however they ferment it. So a lot of them call it SCOBY, these people in the kombucha homebrew industry, and they don't even know what SCOBY stands for. They just, oh, SCOBY, what a funny name. And of course, it stands for a symbiotic culture of bacteria and yeast. Great, so that tells us yeast is present when you make kombucha, and the bacteria that produce the cellulose and acidify the growth media underneath, they are happy to live and be in a symbiosis with yeasts. And so what ZJ investigated was what yeasts are present in kombuchas. He bought a load and did some sequencing of ribosomal RNA regions and found that commonly in lots of different kombuchas, you can find Saccharomyces cerevisiae, baker's yeast, brewing yeast. The yeast, I have a little cuddly bear of over here behind me, and the yeast, of course, we use all the time in synthetic biology. We even have literally synthetic versions of this yeast with synthesized chromosomes. So this is fantastic. This means if we can recreate this symbiotic culture with our specific bacteria that we can engineer and yeasts from the lab that we can engineer, now we can open up growing materials where multiple cells are able to do things. And we know that baker's yeast is very good at being programmed to do things like protein secretion, which the bacteria is not good at. So we applied for funding through a small internal pot, not much money, but we got it. That allowed Charlie in my group and ZJ in Tim Liu's group to be able to exchange between MIT and Imperial. And they both spent 3 months in the respective labs and worked throughout their PhDs on this project together. First thing was how to crack the symbiosis, and a little bit of changing around with different conditions and a bit of reading, we came up with the solution sucrose. So our bacteria can grow a little bit on sucrose, but really not very well at all. And it's certainly not enough to produce any material. It eats glucose and fructose, but not sucrose. It doesn't do that breaking down. Yeast, on the other hand, can break down sucrose. And the way it does that actually is by producing an enzyme called invertase. And interestingly, it secretes that enzyme. So anyone else who's close to the yeast can benefit from the fact that the yeast is secreting that enzyme. We found that if you grow in sucrose with the right levels, very quickly, the ratio of the yeast and the bacteria in the co-culture will optimize to something that allows both of them to grow and to produce the material layer as well. And this becomes a very stable symbiosis over time. So you can take the culture from one thing, grow it again, grow it again, grow it again, and it will always stay at the same ratio of the bacteria and the yeast and produce the material over many generations. This is how it works. We can see some of the yeast cells stained in green here, and the bacteria stained in red with green fluorescent protein and red fluorescent protein expression within them. We can grow the material. This is how the symbiosis is working. And flow cytometry analysis of both the material layers and then the liquid layers as well shows us the relative ratios of the bacteria and yeast within this symbiosis. And we end up obviously with the bacteria being so much smaller and so much more keen to make the material, with way more bacteria in the material layer that's being produced than the yeast. So we had to do some tricks, which we've filed a patent for, to help the yeast be there present in larger numbers and at a higher density at the surface where the material is being produced. And so, for example, one of the tricks we did is we engineer the yeast to express proteins on the outer surface of the yeast cell that like to bind to cellulose, so that the yeast cells are more likely to get stuck up amongst the material as it gets made. And this is one of the ways we can increase the concentrations of the yeast at the part of the growth culture where material is being produced. And we can see here through a bit of microscopy here, we've taken a sample of the material we've produced, put it at an angle, and we're spinning it around and detecting the fluorescence from yeast dotted around the material here. The material got torn in the middle here, unfortunately, in this attempt. So it's a bit of a mess in the middle, but you can see it through the rest of the material. Great, so we can grow the material, and yeast cells are there as well as bacterial cells as the material is being formed. And yeast cells are very easy to program with DNA code to do things. So the first thing Charlie and my group decided to do was see if he can get the yeast cells to secrete something that then goes out and sticks to the material and functionalizes the material so it has a property. Simple thing to do. Engineer a bit of DNA which expresses from a strong promoter, a mating factor tag, which tags the protein that you're going to express so that it gets secreted, and then a protein that you can detect. And so we secreted out beta-lactamase, the antibiotic degrading enzyme. And we made two versions of that, one normal one and one fused to a cellulose binding domain protein. And the cellulose binding domain protein one worked really well. So what I mean here, we grow up a bit of material, we take that material and now we put it in an enzyme assay called a nitrocephin assay. Nitrocephin assay starts with a yellow compound in liquid, but if beta-lactamase is present and active, it breaks it down to make a red compound. And sure enough, our material jellies here, because they've been rehydrated into this assay, the ones that express the enzyme, where the enzyme is fused to the cellulose, they work best. And you get this nice reaction occurring. And this is after growing the material and then washing it a bit. So the yeast cells aren't there and the enzyme shouldn't be floating around. It should only work if it's fixed into the material. Remarkably, this even works with dried material. So we grow the material and then leave it for a week or two just to dry out. It's no longer growing anymore. Cut bits of it out with some scissors, because it's a bit like a sort of a leather paper here. And then take those bits and put them in the assay, and sure enough, they still work. We think the enzyme gets added protection of being surrounded by cellulose as well, so it lasts longer than it would if it was just on its own. Even after 6 months, we're still seeing activity of this. And so we can swap out beta-lactamase for other enzymes and make other things. So we can break down XGAL with MEL1. We can break down ABTS with lacases and get the color change there as well. Pretty cool. So there's lots of uses for that. You can think about growing material filters, which have the enzymes embedded in the material and made as the material has been grown. ZJ took this to another dimension by having the yeast cells produce not 1, but 5 enzymes at the same time and secrete those out in one go. And those enzymes all being cellulases, so that they act upon the cellulose. They break it down and weaken it wherever the yeast are present within the material. And we saw through materials testing that we had weakened the cellulose significantly. You can see here the tensile strength being halved in the presence of the yeast cells in that area. And so potentially you could think about growing this and having this system triggered in certain areas of the material as you're growing, so that you end up with a material which has patches where there's a stronger material and other patches where it's weaker. So you, for example, you might be able to, in the future with Turing patterns, be able to have stripes of weak and strong regions in the material as it grows, and that will be self-done through DNA programs. The other thing we can do, which we do a lot in my group with yeast, is engineer the yeast cells to sense something and report on it. So we call that like yeast-based biosensors. And so we can literally, in our system here, take yeast cells that we've built for other projects to do sensing and just mix them into our SCOBY, this symbiotic culture, and that will grow us these materials which now contain yeast cell biosensors in. And so here's an example here with an engineered sensor for the hormone beta-estradiol, which is a significant problem in municipal wastewater due to people taking birth control pills. And that, in response to beta-estradiol, will produce GFP signal. And so we can then grow these little bits of material that when you then take them and move them into water that contains beta-estradiol and let them grow for a little bit longer, maybe a few hours, they will start to produce green fluorescent protein. Intriguingly, this can even be done after dehydrating this material and storing it for months. So we grow one of these material layers with the sensor yeast cells within it, just let it dry out on the bench. It becomes this sort of little paper disc. You can put that in a tube and store it at room temperature in the fridge for months. And even after 4 months, if you then take that disc, put it in your water sample, add a little bit of sugar and incubate at 30 degrees for a while, you'll start to see fluorescence turning on if the pollutant you're trying to detect is present. And our aim on this was to make a system even better, where you can have a system that senses a pollutant, and then instead of producing just GFP, it can produce an enzyme like a lactase, which breaks the pollutant down. And lactases are known to be able to break down beta-estradiol. And so this cell could potentially do this, although in the case where we've shown it here, we get it to instead break down a colorful dye rather than the beta-estradiol. But it only does that breaking down when the beta-estradiol is present. So great work there by Charlie on this. The other thing ZJ did over at MIT, which kind of really excited me, very visual, was engineer this system so that these cells, as they grow and they make the material, can respond to light. And so this is very cool. It allows us to use optogenetics, where you shine light in certain areas onto cells to be able to get the materials to have patterns on them based on what light you've been shining. So ZJ took a system described by others and got it working in yeast, where 2 halves of a transcription factor come together in the presence of a light being shined upon them. And that then leads to triggering expression of an enzyme that is secreted from the yeast called nanoluciferase. And that luciferase, when a chemical reagent is present, will shine light back. So it's bioluminescence, right? He made 2 versions of this. The one where the luminescence diffuses, the luciferase diffuses from the yeast, and another one where it sticks around the yeast. The one where it diffuses gives a slightly brighter signal, but the one where it sticks on the outer surface gives a slightly more defined signal as well. And so with this, we were able to achieve something that I long wanted to achieve in synthetic biology, which is you may have seen many people demonstrate, shining light onto cells on a Petri dish, and then those cells produce something like a picture, smiley face, or a logo in response. Great, that's been done many times. I wanted us to be able to do that, but also make the material as well that the picture is shown on. So this would be the kind of the equivalent of the Polaroid camera, where not only do you take the picture, but you also get the material with the picture on it. And sure enough, we did this, shining the MIT and Imperial logos onto the material as it grows, and then having a bit of material that you can then take into a dark room and it glows luminescence back to you, showing these logos. The other thing I learned in this process is that MIT has a much more friendly logo for trying to do optogenetics experiments than Imperial College's really complicated logo, which is much harder to get correct. So here, just to summarize, we see yeast as the specialized cell within the material. If I think back to that analogy at the beginning of plants, we see in plants within the tissues many specialized cells that contribute something to the material property of that part of the plant. So for example, in a leaf, there are specialized cells at the edge that do things like secrete out a biopolymer, like a wax, that gives you a waxy surface that changes the properties on that side of the leaf. Others will be doing sensing, responding to light, producing molecules that benefit the rest of the cells within the material. And so here we sort of see this engineered synthetic symbiotic culture of bacteria of yeast as enabling the bacteria to just do their job of producing the bulk material. And then every now and again among the bacteria, there's yeast cells that are doing specialized tasks, secreting enzymes, adding extra biopolymers, responding to light. And we published this in Nature Materials at the beginning of the year, and it got a lot of interest because there's a lot of people thinking about living materials, changing materials in industry so that we don't use plant-based materials like animal-based materials like leather and instead move to plant-based materials. And also a lot of people who just love kombucha and stuff to do with home brewing of kombucha who want to do this kind of stuff themselves. I will say though that we are focusing mostly in my group now back on the bacteria, where people like Marcus have made some really fantastic recent progress. For example, putting optogenetic systems into now the bacteria so that we can do this same pattern formation through light, but now not having to rely on those yeast cells getting it also into the bacteria. So triggering protein expression in response to light. And this now, because the bacteria, there's so many more of them and they're so smaller, gives you much higher resolution. So we can do some really remarkable light-induced patterns into this grown material. This is a piece of material the size of a Petri dish. And also we have a way where, depending on how intense the light signal is, we can get different amounts of expression. So we can really tune the production within the bacteria of proteins in response to different intensities of light as well, which is very exciting. So summary, you're probably thinking, what are the things we can use this for? Well, we know already that bacterial cellulose is used in industry in bacterial separators, surgically implanted materials that go into your brain when you have brain surgery, for example. It's used as scaffolding in tissue growth from stem cells. And people are looking at making active wound healing materials. Bacterial cellulose is safe to eat. It has no toxicities if you put it into you. It's really a very good material to work with for lots of different things. And yeast, of course, is also safe to eat and doesn't also have endotoxins like E. coli does as well. So we think yeast can work very nicely within these marketplaces here. We're looking at immobilized enzyme materials based on some of the work we've done, and we want to make sense and respond living materials. And of course, there is this incredible thirst for replacing animal products with plant-based products. So leather replacement and creating other new textiles is something of particular interest. And so we have in my group collaborated with a designer who works in the fashion industry called Jen Keane. And she has developed this very unique way of growing the bacterial cellulose with threads forming weaves and then can grow bacterial cellulose-based shoes using her approach. And we even have the ones that produce their own color as they're growing. The world's first, I guess, item of clothing that grew the color through genetic modification so that the dyeing process happened due to GMO as the material was being made. And Jen, plus others from ex-members of my group, have formed a company based on this and are getting significant attention. That company is called Modern Synthesis. So thanks for letting me talk to you for an hour about this. I can stay for a bit longer for all sorts of questions. I would like to particularly thank all the people in the materials side of my group who've worked on these projects over the years. Viv, Charlie, Marcus, Wolfgang Ott, who was with us briefly, ZJ over at MIT, who came to our group for quite a while, Michael Floria, the student, amazing student who started a lot of this off before going off to Harvard, and the iGEM team that got us going on bacterial cellulose 6-7 years ago. And then also thank my collaborators, Kunyang, still here collaborating with me at Imperial on the material side of things, Tim Lu, at MIT, Jen Keen at Modern Synthesis, and other people in very exotic places like NASA and University of the Arts London. And then most obvious, I also have to thank our funders, particularly the Engineering Research Council in the UK and Imperial and MIT for the starter grant they gave us. And thank you for listening. Hopefully there's lots of questions that I can entertain you with.
Thank you, Tom. That was amazing. I couldn't stop listening to all this amazing stuff you are doing, all these materials created from the microbes, and it's really amazing work. And so now we have time for the audience to make questions. Either you can write the questions in the chat or ask me about open your mic, and you can also by yourself make the questions. So in the meantime, I have a few questions that I'm interested to, because it was for me curious to know why. I don't know if you have an answer, because you are not interested in the biology of this Comatella K. reticus, but it's really amazing why they produce this cellulose. I guess it's kind of protection in the nature to create an environment for them to protect and grow the, I don't know, what do you, what's your hypothesis about that?
Yeah, that's true. I mean, so some of the people I've hired over the years, like Viv Hussens, who you see here, come from working with other bacteria and have a lot of expertise in biofilms and biofilm formation. So she came, joined our group and went, it's a biofilm, it's biofilms. Why are they so stressed that they produce biofilms all the time? Why are they so stressed that they produce biofilms when you give them huge amounts of sugar and make the conditions perfect? So that makes me think it's like a biofilm that's become useful at some point. So biofilms are normally there to protect in times of stress, maybe protect. So maybe it's useful against UV, maybe in the natural world. This is predominantly found like on the skins of fruits along with yeast, where there's lots of nice sugars available, but there's also strong sunlight, maybe like on grapes, somewhere near Valencia, maybe with nice wine. But I'm not sure about that, because there's lots of other things you could do to prevent against that, and biofilms only really happen when it's stressful. So why does it do it when it's not stressful? So my feeling, and it gets debated quite a lot with people who work on bacterial cellulose, I fall more into the camp that it's a mechanism to outcompete others. They can basically seal off the growth, the liquid with sugar in it, and prevent other people from coming and contaminating it, because they very quickly make this very top layer, and they make it right at the top at the air-water interface. And that's, for example, that seals the yeast underneath, and that's why the yeast start fermenting more, and that's why kombucha is, fizzy and all of those things, because you get that fermentation. So I think what happens is this is their way. They spend 30% of their glucose because it allows them to get rid of competition,
right?
By preventing other things from the air falling down and getting underneath.
Yeah, must be something like. But do you know if they are in the nature, not in the kombucha, but in the nature they are in the, I don't know, where they are isolated? In the solid surfaces, or more in the water, or?
Yeah. No, it's a good question. So, most of the strains people work with have been got from people who do kombucha, right? So you go, there's a community, people who brew kombucha for some reason all over the world, right? It's popular in India, popular in Japan, popular in Korea, has been for a long period of time. And so most of the strains people work on are ones taken from people who've worked in kombucha or worked on vinegar production, and then they've gone, okay, well, these are used in this industry, let's do them. So there's not as much information about what's actually out there in the wild in terms of genome sequences. So the story, actually, let me go all the way back, about us and our genome sequence and working out what our strain was is kind of a little bit fun. Let me go all the way back to that. So Michael's sequence, so we got our SCOBY from a company based in the UK called Happy Kombucha Limited. a little, you know, someone who just runs a company from their house that sells these scobies to people to be able to do home brewing.
Okay.
And make their own, right? Because it's a trendy thing, right? You can literally buy that thing on Amazon and get it delivered to you, and then start doing
okay.
it. So we
noted.
we took that, we isolated it, we then genome sequenced it, and we were a bit puzzled because it came back as something to do with Raticus, whereas everyone else in the field was Xylanus and all of these other ones that were. And if you read in the text, they were all from kombuchas from Korea, India, Japan. And we're like, Raticus? Where? So we searched these other ones that had partial sequencing, and they were isolates found on like spoiled apples and cider production and things like that in kind of North Italy. Mm-hmm. Like, okay, that's a bit weird. Well, why do we have, what's gone wrong here? Because we're doing ours from a kombucha. And so we called up Happy Kombucha and said, oh, do you know where did you get your thing from to start this? We thought you'd got yours from people in India or Korea. And they said, no, Happy Kombucha is a family thing where for over a century, our family were based in Czech Republic near Prague, and we grew kombucha style drink. Almost no one did, but we did it for some reason, and it's been going in our family for over 100 years. And in 1960 something, when the USSR came in to crush the rebellion in Prague with their tanks, we fled to the UK and we took kombucha with us because we had to, because it was the family tradition. Mm-hmm. Yeah. And then we're like, okay, well, Prague is not that far from northern Italy. And okay. Sure enough, Raticus is this ancient kingdom from hundreds of years ago that covered this area of Austria, northern Italy, and some of Czech Republic as well. So it's something that they've probably isolated from apples and things and fermented with and made something like kombucha. They were like, oh wow, you've done some genome sequencing heritage for our business. So it was a fun story.
Okay, so I recently have seen that they discovered the evolution of the apples were originally from Asia, and then they spread across the whole Europe. So I guess the symbiosis or the originally the first origin was Asia, and then they spread all around the Europe and then America.
Yeah,
associated to the apples. I don't know.
Yeah. If you think about it, when you buy that like fresh apple juice from a farm, and it sometimes it gets, you know, you leave it a bit too long, it gets quite lumpy with like some things floating around on the top, and you go, oh, is it safe to drink still? That's probably relatives of these cellulose producing bacteria.
Okay, that's nice. Javier also said a comment that the fact that the bacteria is protected prevents also the sugar to be consumed by more efficient fermenters like the yeast.
Yeah.
What do you think? It's also like a metabolic adaptation?
Yeah. I think what's interesting though is what do the yeast get out of this relationship? So we call it, everyone calls it symbiotic culture of bacteria and yeast. Symbiosis is a two-way arrangement, right? Mm. And we know that the bacteria get the access to the sucrose. What are the yeast getting in response? I've asked other people and everyone says, well, maybe it's not symbiosis. Maybe it's the yeast are getting held back. But it can be that I think the yeast get the benefit of having the material, the liquid underneath them to themselves because it gets sealed off, and yeast is very good at consuming sugar when no oxygen is present, unlike a lot of other microbes.
Yeah, I guess it's two-way.
Yeah.
So another question is, Amparo Lopez congratulates for a fantastic seminar and says, if you know if there is any way to continuously produce the bacterial cellulose.
Yeah, so continuous production. So we do, I guess, what we would call batch production. We will take a flask, get it in, leave it sitting there, get the material will start to appear after 3 days. And if you want a big thick layer, you might wait 2 weeks and then take that off. And if you were to wait a month, it doesn't really get that much better because you've started to exhaust everything. Samsung, who want it in high yields, they stir and ferment. So it never forms the material, it forms more the polymer, and then they take that and form it into the material themselves later.
Okay.
There are groups that have developed lots of cool things, such as like these sort of mini conveyor belt systems, you know, a bit like an escalator or whatever, where it grows stationary, and then as the material layer is produced, it's something's grabbing onto one end and slowly pulling it off.
Really?
And so it gets continually produced whilst, and then over here more is getting produced, and others have produced these sort of drums that slowly turn around.
Okay.
So you get layers and then more layers and more layers. So you can continually produce it with some creative
continuous systems, right?
Yeah.
Okay, and she also asked if it will be economically feasible for active food packaging applications, because in our institute there are people working on packaging, and they will be interested also in this material to be used as a packaging.
Yeah,
material.
and I have seen, you know, artists conceptualize that we should replace plastic packaging in food with this cellulose from bacteria, grow it, and it can be reused a lot better because it's kind of waxy. It's a bit like a waxy paper bag or something like that, right? And so it's quite nice for reuse, and it's biocompatible, and it will biodegrade over time. So yes, you can, and you could think about putting in those yeast cells to respond to things, like respond to the signals that are given off when meat goes bad or toxic, or, you know, maybe in the wine industry, you could have response to the molecules which show that the wine has been corked, it's gone wrong and gone bad or something like that. And then you could have a color change, so you know, there's those kind of things. Question then, the other side of it is the economics, which is interesting. This is a very cheap material to make at scale. So the food industry that make this dessert product called nata de coco in the Philippines, Vietnam, Indonesia, they will sell you a metric ton of bacterial cellulose for like $100.
Wow.
Okay. And so the majority of what you're paying for is someone's got to put that thing on a container and send it through the Suez Canal and get it to Europe. Because I know that because a colleague of mine bought a metric ton of it, and it was very expensive to transport, but actually to buy was very cheap. Now the issue is that sounds very cheap, and it is made at huge scale in nata de coco factories, well, which are actually just farms. It's just in a room in someone's farm. They'll just grow it in vats. So there's the scale, and it's cheap. The problem is that's in the hydrated form. And so as soon as you want to get it down to something like paper or leather, well, that's 99.9% of the weight evaporates off. And so now you start to look at the price, and you're like, okay, I get a kilogram, or I get 5 kilograms for $100. Okay, that's starting to get expensive, considering paper that you buy to put in your printer, you get a kilogram of it for a few dollars. You can see then the economics aren't quite right. So we don't see it as an obvious, like, okay, if everyone just switches to bacterium-made cellulose, we can abandon cutting down trees for paper. But the economics are probably good for, you know, trying to make something fancier than paper, like paper that can sense and respond to things.
And also maybe it needs another kind of polymers to increase resistance to be used as a packaging.
Yes and no, because you can really get different material properties depending on how you treat the cellulose as it dehydrates. So you can dehydrate it and you can mix in certain other polymers during that process and get things to be flexible. You can wax it. I mean, ultimately, if you think about you dry animal hide out to make leather, well, leather, suede, all of those different products related to leather, they all feel slightly different. And that's really just more about how they've been treated in the final stages, whether they've been oiled or waxed in certain ways. And it's exactly the same with the bacterial cellulose. You can get it to be very tough or flexible or whatever, just depending on how you treat it in the final stages. Now, some of those treatment methods are going to disturb things like how cells respond or how enzymes present in the material that you've engineered behave. And so that's a complex thing that we have to consider with future applications. Which enzymes can you put within the material that will still function when the material is dehydrated and waxed, for example? And so we might need to move to, for example, only using enzymes that come naturally from thermophile bacteria, because those will be more stable in low water conditions and high temperature conditions, for example.
Right. So Sebastian want to ask about optogenetics. It has the potential to create complex metabolic flux to produce biotechnological relevant secondary metabolites, and also the opinion on the future of this technology.
Of the optogenetics?
Yeah.
So for us, the optogenetics, we want to use it. So this is the yeast-based one. We want to use it to define patterns in the material as the material is growing. So for example, one thing we've not been able to achieve, but we tried quite a bit, but people have moved on, so it's on pause at the moment, was if you shine light in certain areas, we can get the enzyme here that produces light to be secreted. Why not instead shine light here and have the enzymes that break the cellulose down secreted there? Okay, and so that would enable you to, as you're growing the material, you could shine beams of light in certain areas in the material, and those areas, the cellulose will be weakened in those areas. And what I really wanted to achieve, which we've not achieved yet, but would be a nice one, is you shine a template onto the material as it grows. The material is weaker in that area. Then as you leave that out on the bench to dry, it will fold at those weakened points, and then it will fold into a 3D shape. Like really? It will spontaneously do origami.
Okay.
I mean, so stuff like that has been achieved not through genetic engineering, but through weakening things so that as they dehydrate, they fold up on themselves and make a little structure.
Mm-hmm.
But wouldn't it be amazing to be able to shine a light and have the DNA and it all forms some structure in response? And so that's one of the areas we want to use it for, because light is very good way to define some complex patterns on things. Mm. Now, a bit more cynical, we could just like shine company logo onto it, things like that, right? So you could grow materials for a company where they can say our logo has been genetically printed onto the product or something like that, right?
Yeah, it looks promising, really. Another question is about acetic acid bacteria. José Manuel Guillemot said he worked with them for a while and they were very difficult to grow. How reluctant are they to be transformed in plasmids or other DNA material?
It's difficult. Everything we do with the bacteria is a lot more hit and miss than in the yeast and in E. coli. So although we're doing some good work here, I'll just go to the end slide. There's a lot of, okay, it didn't work. Why didn't it work? Not really sure. What really matters is getting the conditions right so that you grow the material when you need to. So it might be, getting the starter culture right. We tend to because it's so hit and miss whether a culture will kind of pick up and take off and grow the material, we tend to very rarely go from a single colony on a plate and grow that and produce. We'll do that sometimes. We obviously have to do that when we engineer it, but then once we've done that, and we'll pick a whole load, and a few of them will be established and grow well and produce the material. And then from that point onwards, every time we want to use those, we go to the material, cut some of it, and use it to seed the next culture and then the next culture. For some reason, these bacteria are very hit and miss when they're on their own. But once they've started to form this community, then you can replicate that reasonably well. We do find that the bacteria are easier to work with if the yeast are present.
Okay. So I have a question regarding the symbiosis between the bacteria and the yeast, because I guess the bacteria grow first, and then when the yeast start to grow and produce ethanol, the bacteria is just stop, and there is like a not parallel growth, but it's a kind of competitions until balance is established, right? So I was wondering if you are interested in use another yeast that doesn't produce as much as ethanol as Cerevisiae, or you don't care about that, or what do you think about this competition and balance?
Yeah. So your hypothesis is wrong. The bacteria produce the cellulose better if ethanol is present.
Oh, really?
We don't know.
They don't care about ethanol.
Yeah. So if you have the bacteria only and you want to produce a really large amount of cellulose, then people have experimented on this for years, and the company that some of my students formed did research on this as well. And so some of the things you can do that somehow get the bacteria to produce more include ethanol. That somehow that triggers them to produce more. Produce, I've forgotten the name of it, but the molecule that makes the smell of bananas.
Okay.
It's a volatile molecule that triggers even more production. Stuff from coconut juice. If you go to the shop and buy one of those coconut juices, if you pour coconut juice water in as part of the growth media, boom, you get like a huge increase in yield. So there's all of these things, and it must be related to their natural environment, that they like to produce more when there's fermentation going on and interesting molecules being produced.
Stressed.
Yeah, well, stressed or symbiosis with yeast, maybe. Who knows, right? We haven't upped the ethanol to a point where it stops production, but I think it's fine up to, with 3, 4, 5%.
Okay.
It helps.
I see. Yeah. So yeah, it's an interesting balance between both of them. And we're using other yeast like I don't know, other yeast that are more protein producers or produce more other components.
Yeah. So the go-to protein secretor in biotech for yeast is Pichia pastoris.
Yeah.
So we have tried that as well. It's not as good a relationship with Pichia pastoris, because Pichia pastoris does not like it doesn't have the Crabtree effect, which is the ethanol production and things like that. You can get that co-culture to sort of work, but it doesn't work as well.
Okay.
But really, Pichia pastoris is very good at protein secretion at very high yields, less so because each cell is so much better at secreting protein, but more because that yeast you can grow at incredibly high densities, so that there's really, really loads of yeast secreting a little bit of protein.
Okay.
And that really, if you think about the balance of a material, you can't go, you don't want it to be just full of the Pichia.
Okay.
Although Pichia is good at protein secretion, it won't provide that much of a benefit to move to Pichia as opposed to Cerevisiae.
Okay, fantastic. Tom, I think it was a really incredible talk, and we have enjoyed a lot to see
Thank you.
how these materials are evolving and how powerful they could be and useful, and
yeah, I think
to apply to.
Yeah, worth saying as well. It's all stuff we eat, and we're all happy to eat. So don't be afraid if these materials start to appear in the shops one day.
I hope. Yeah, I'm also noticing that other people. It's congratulating you for your fantastic talk. It was really a pleasure to have you here, and yeah, thanks. I hope in the next future you can come to Valencia in better conditions to enjoy, and we can have you here.
That would be nice. Yeah,
that would be nice. But maybe next year, if we have opportunity, we can meet or meetings or somewhere.
Yeah. Likewise, everyone.
Yeah, thank you.
Come to
much.
London.
Yeah, we will go also. Thank you.
Bye-bye, everyone.
Bye.
Thank you for listening.
Thank you.